talk about antibiotics a little bit later. There are a few that work on things like translation or transcription, but far and away, most of them work on cell walls. Cell walls are important, but also they happen to be on the outside of the cell, so your antibiotic doesn't have to get as far to actually work. All right, so they're specific, right? That's another sort of entity with why cell walls are such good targets for killing microbes. Again, peptidoglycan, right, is not something we see in eukaryotic systems, and so it's not going to impact us if we take an antibiotic for microbial cell wall. Yeah, you get rid of the cell wall, cells tend to lyse. It's generally not very good for a cell. And yeah, again, not having to permeate that cell membrane can be really beneficial. Okay. All right, so we've talked about sort of that peptidoglycan, essentially polymer. A big important aspect of this, right, are these interlinkages, right, that you have between the polymers. Essentially, this makes sort of a cage-like network. And essentially, that cage-like network, you have multiple layers of that. There are still gaps, right, between these molecules, so things still do diffuse through. So it's not acting as a barrier to, say, small molecule diffusion or transport or anything like that. But really, this is a good structural support here. This is a little bit more what it looks like in real life. So it's not necessarily sort of perfectly lined up cage, but sort of more a dynamic structure here. And again, these molecules kind of wrap around and around cells. So this is just kind of an overview of how these peptidoglycan chains run. I'm sorry. And these peptidoglycan chains essentially, sort of span out towards the poles of the cells. And you can imagine for the way that a rod-shaped bacteria divides by binary fission, this is a pretty smart way to do it, right? Because you can just sort of internally keep adding sort of peptidoglycan from the center of the cell. All right, so we're going to talk a little bit about lysozyme as well. So isozyme is an enzyme that breaks down beta-1,4 glycidic bonds, specifically between N-acetylglucosamine and then muramic acid. And so what happens when you break those bonds, right? So chewing away at those essentially cell wall, right, breaks down that sort of structure that you see. You get water entering the cells, and eventually, with enough sort of osmotic pressure, you get lysis of the cell. This, of course, depends on the solution that you put them in. So you can, for example, do things like put them in an isotonic solution, so a solution that has a similar chemistry to the inside of the cell, and you wouldn't necessarily get this sort of lysis, but most solutions are going to be very low concentration compared to cells. All right, so we touched on a little bit, right, how antibiotics tend to focus on cell walls. The classic case of this is being penicillin, right? This essentially blocks production of the transpeptide bridges between these polymers, essentially your peptidoglycan polymers. So essentially, you have enzymes called transpeptidases that that essentially penicillin inhibits. And essentially, it prevents sort of that cage-like structure. So you can still have the polymers, but you don't get sort of that cage-like structure. All right, so imagine you are a cell, right, and you are just exposed to this peptidoglycan, or sorry, this penicillin, and what happens, right? So eventually, essentially, okay, we'll move on to this. So eventually, right, it'll again do a similar thing to lysozyme, right, and force cell lysis. But what's sort of a caveat here? So penicillin only works on bacteria that are growing, not growing, gram-positive, gram-negative. couple people for gram-positive, and a few for gram-negative. Okay, so this is one we should probably discuss. So think about why penicillin would work preferentially on a gram-positive, gram-negative, or why it, nobody said not growing, right? So you could rule that out. But why might you pick gram-positive, gram-negative over something like growing in general? So you give a few minutes to talk about it. We'll come back.
Versus the gram-negative, you have the cell membrane. So that cell membrane is going to be blocked. But then I don't know at all how you
as the answer. What was your logic?
I said in non-growing cells, cross-linking doesn't happen, and so penicillin will have no effect because it directly degrades the cross-linking process.
Okay. What about somebody who picked gram-positive? Gram-negative? So is everybody on board with growing now is the answer? Is that the, okay, well, let's re-poll real quick and then. That's what I want. Alright, so nobody actually changed their mind. There we go. Alright, so gram-negatives moved over to growing versus, okay, so, alright, any brave gram-positive person want to speak up? Or, sure, no? Okay. So the answer is growing, okay? So, yeah, I know. But right, so the idea is, right, so if you're a cell that already has transpeptidase bonds built, right? That you're essentially, like, if you're not growing, you're not going to be building more of those, and so penicillin is going to do nothing to you, right? Gram-positive versus gram-negative, right? Both of them have peptidoglycan, right? Both of them use it as that structure, that, integrity, right? So even though gram-negative have less and gram-positive have more, they're still dependent on peptidoglycan, right? So something like this transpeptidase inhibitor still impacts gram-negative versus gram-positive bacteria. Okay, so there are some, clinical application issues, right? So the amount of concentration that you have to add to kill a gram-negative bacteria versus gram-positive might be different. Certain types of cell wall inhibitors might not actually get through the outer membrane of gram-negative bacteria, but penicillin does.
So we were confused on the basis that if it's growing, does penicillin inhibit the bonds or does it degrade
inhibiting those sort of transpeptidase bonds formation. So essentially, you only are getting sort of the cable-like structures, but there's no integrity kind of in between the cells. So more or less, kind of what happens with lysozymes, even though there you're just impacting that 1,4 bond, right? Now you're essentially doing, you still have those transpeptidase bonds with lysozyme, but now you don't have the ones in between the polymers, and you still get more or less the same effect, which is you can get sort of leaching out and rupturing or lysis of the cells. But yeah, so it's not actually breaking down the bonds themselves. So if the bonds are already there, it's not going to impact the bonds that are already functioning. So that's why it's not going to work on a non-growing cell, for example. But that making of new bonds is going to be the problem. Does that make sense? Okay.
So for like a mature gram-positive bacteria, it would have no effect on it, right?
Yeah, so if the gram-positive bacteria was not growing at all, would not impact it. Yeah. Yeah, so it only would, and so this is actually one of the reasons why we take antibiotics for such a long time for some things, right? So I don't know, a lot of the cell wall inhibitors in particular, sometimes you might have a 10-day course of antibiotics, and that's because you can have some of these latent cells that are still kicking around, and if those kick into a growth phase 5 days later, right, you'll just get that infection back again. Right. All right, good job. Okay, so beta-lactamases are enzymes secreted to break down beta-lactam constructs. So again, things like penicillin are molecules called beta-lactams. They sort of, again, they're this competitive inhibitor of things like the cell wall transpeptidases. And so if microbes have essentially enzymes that specifically target those sort of beta-lactamase compounds, you can actually break down things like penicillin. And so antibiotic resistance essentially are genes that help you sort of deal right with antibiotics. And this is sort of a big class of antibiotic resistance genes, are molecules within the beta-lactamase. All right, so, and these are problematic in terms of antibiotic resistance because now you maybe not are completely just specific to penicillin, but maybe anything that has this sort of beta-lactam ring. And so there are different types of antibiotic resistance, and we'll talk again more about this later in the class. But one of the big challenges, right, is having these sort of broad spectrum enzymes that get passed around across strains that give you sort of inherent resistance to a bunch of different types of different antibiotics. All right, so probably why a lot of you, gut instinct maybe said that gram-positive bacteria are going to be more affected by something like penicillin, that they have about 90% more, right, peptidoglycan. But again, they're still both utilize this as sort of integrity structure, and some of these differences probably just have to do with the environments that these organisms live in, and or the conditions that they deal with. But I guess one thing to kind of keep in mind is that even though, for example, you don't necessarily have a membrane out here, there are a lot of similar sort of functional roles that the surface of cells have to deal with. And so these cells groups just have different ways of accomplishing it, right? One being that they have these incredibly long sort of teichoic acids that actually span from the membranes to the exterior of the cell. So we'll talk about in gram-positive bacteria where they have sort of these LPS moieties, so they still have lipid interactions between that surface of the cell and what's going on inside. You also have proteins that are associated with the cell wall in general. So again, slight differences, and overall very similar sort of charge structures or charge bases. So you still have a predominantly negative overall charge. All right, so again, the wall is similar to, it's not an impermeable barrier, right? Again, so it's not really going to be a blockage for diffusion of small molecules. So a lot of things can freely diffuse. These are actually relatively large spaces or gaps in between there. Probably not everything is going to diffuse through the cell wall, but it has, most of the nutrients that the cell might need are going to freely diffuse, most of the carbon substrates, etc. All right, so gram-negative bacteria have a slightly different, outer orientation, and I would argue that, part of having this sort of outer membrane structure does give them a little more surface diversity than organisms within the gram-positive bacteria. And so a lot of these, the molecules and what's going on out here tends to vary quite a bit between gram-negative and gram-positive bacteria. But one thing, I sort of mentioned is that this is not as important a barrier for solute diffusion as the cytoplasmic membrane. You can imagine if you had to do active transport at two membranes, right, that would be incredibly energy costly. And so a lot of cells actually have structures that allow at least diffusion of most nutrients that the cell would need. So, kind of similar to that idea of diffusing through a mesh, you can still penetrate that outer membrane if you're something like a nutrient. All right, so again, membranes have a lot of kind of extra ability to do structure, and essentially there's a lot of protein associated with this outer membrane in particular, but also different sort of polysaccharide entities with things like LPS. But the periplasm then also becomes this kind of important space. It houses that cell wall, and it also kind of helps protect, a lot of sort of the important stuff going on at the inner membrane as well. All right, so, you have two outer membranes, you have the outer membrane here, you have two membranes with an outer membrane that acts as more or less a barrier to harmful compounds, potentially heavy metals and those sorts of things. But it also is a main surface area for important interactions. And so a lot of organisms have sort of specific structures that are involved in signaling. So the LPS has a lipid moiety here, a sort of core polysaccharide, and then sort of an antigen-specific polysaccharide. And so this is something that, again, tends to be sort of a unique fingerprint to a lot of gram-negative bacteria. And this, is, again, kind of a general structure, but little differences in, your LPS essentially can impact, right, how your immune system, for example, sees that particular molecule. So for a long time, we taught about these LPS structures as being things like endotoxins, right? So when a cell lyses, your immune system sees these molecules and essentially you react with something like diarrhea. But that's not every single, lipid A or sort of LPS structure that you would get, right? So those are ones that your immune system is particularly tuned to identify as foreign or bad agents, right? And then they sort of signify some level of infection and your body reacts accordingly. A lot of your commensal organisms, hold that thought, a lot of your commensal organisms also have these exact same structures, right? And your immune system responds very differently to the exact same molecules. And in fact, it responds in a very healthful way. And it turns out that lack of exposure to some of these molecules, especially early in your development, can be really, really bad. So these are generally important, but the details here, which vary across different species of bacteria, make huge differences. All right, sorry.
Okay. Are there any proteins that exist that will connect the inner and outer membrane, or does nothing really ever go across the periplasm to connect them?
There are proteins that, at least loosely associated, can span, for example, the peptidoglycan and connect the two. And that organism Shigella that I mentioned, I think the first day of class, where they do extracellular electron transfer and the electrons get to the outer membrane, and they essentially have proteins that do just that. So, essentially they have a molecular wire that spans from that inner membrane to the exterior of the cell. But it's not, as far as I know, it's not terribly common for that to happen, but yeah, there are certain cases where you can have a protein network that spans all the way outside. I guess another example of one would be, we'll talk about like type 3 secretion systems, things like pili. A lot of those are rooted in the inner membrane, but might span multiple, across to another cell or multiple cell lengths even. Okay. All right, so again, endotoxin is an example of one of these sort of LPS structures that in the case of pathogenic organisms we respond very badly to. All right, so in terms of archaeal cell walls, there are some sort of similar, there's a pseudopeptidoglycan type structure that some archaea do utilize. Most of them do not use there's no traditional peptidoglycan, but there's like I said, sort of a pseudopeptidoglycan that uses a beta-1,3 linkage here. This makes it insensitive to lysozyme. It's also going to be insensitive to antibiotics, so sort of traditional cell wall antibiotics. So currently, we know of no archaeal pathogens, which is great because we don't really have antibiotics for archaea either. But so far, we don't know of any that are pathogenic. So what's more common is that archaea cells tend to have a prognathous cell wall. So they make something called an S layer, which is sort of a prognathous shell that's on the outside. So this is S layer or surface layer. These tend to be made up of sort of self-assembling proteins. So essentially, you express these molecules and they assemble into these sort of grid-like lattices that sort of sheath the cell, which are pretty cool. So they form a really nice sort of crystalline structure. Again, these are gonna have big enough holes that you can get transport or diffusion of important molecules to the cell themselves. But they're, again, still these sort of highly ordered and protective arrays. Some bacteria also have S layers, and we'll talk a little bit about that next, which in sort of alternate configurations. So, yeah, gram-positive and gram-negatives are by far sort of I'm not gonna say the most abundant, but the best studied and the organisms we know the most about, right? And so this is commonly the way we think of at least bacterial cell structures. Most of the archaea that we study have S layers. Again, there's a few exceptions there. But yeah, so there's a couple of different examples, and we'll talk about these examples on Monday. Just as a quick reminder, next Friday we have a paper discussion instead of traditional lecture, so you won't have pre-class questions, but I'll ask you to read a primary literature paper and come ready to discuss